He kōna I purangi tēnei nā te reo irirangi o Aotearoa.
1/4 of the world's population is currently infected with the bacteria that cause TB. And we still have over 1 million people per year that die from tuberculosis.
Kia ora, nau mai haramai ki te ao hurahanga. Hello and welcome to Our Changing World. Ko Cleggan Cannon tēnei. We don't hear much about tuberculosis in New Zealand because the bacterium that infects humans isn't really spreading in the country anymore. But as Dr. Kerry Hilligan of the Malaghan Institute of Medical Research points out, tuberculosis, or TB for short, is still the leading infectious disease killer worldwide.
It was briefly overtaken by COVID-19, but we now have that more under control, and it's re-established itself at the top as the leading cause of infectious disease death.
Some New Zealand farms have issues with bovine tuberculosis. But if you're talking about people, Health New Zealand says we see approximately 300 cases per year.
Pretty much all the tuberculosis cases we get in New Zealand are imported. So they've come in from people who have moved from overseas. There have been a few small outbreaks of TB, generally in settings where people are in close contact. So for example, in a prison in the Waikato, there was a small outbreak of TB. But in general, the rates are extremely low in New Zealand.
When we look back in time, across human history, tuberculosis has always been there. In ancient Greece, Hippocrates described its characteristic lung lesions and symptoms. In the Middle Ages, it was called the king's evil, that could be cured by the royal touch. In the 18th century, it was the robber of youth in Europe, known as consumption because patients simply wasted away. Pulmonary TB infects the lungs, causing persistent coughing, which is how it spreads from person to person. And while it usually targets the lungs, it can affect any organ in the body. In the 19th century, estimates suggest it killed 1/4 of the adult population of Europe. Which is why two French scientists, Albert Calmette and Camille Guérin, turned their attention to figuring out a way to stop it. It's 1902 at the Pasteur Institute in Lille, where tuberculosis is rife and deadly. Calmette and Guérin take a swab of bacteria from the udder of a cow infected with bovine tuberculosis. They figure out that they can culture it. They can get it to grow using a combination of potato and ox bile. Every 3 weeks, they subculture it. They take some of the growing bacteria and streak it out onto a new pile of potato and bile. And they inject some of it into a cow to see how sick the cow gets. And then they do that over and over and over. Streak, grow, inject, sick cow. Streak, grow, inject, sick cow. Streak, grow, inject, slightly less sick cow. Until 11 years and 230 subcultures later, they inject the cow and it doesn't get sick. They've tamed the bacterium. It's still Mycobacterium bovine, it's still alive, but over this process it's lost its disease-causing abilities. It's now an attenuated bacterium. This is what they've been hoping for. The bovine version is quite similar to the human one, so they now have something they can give to people that will prime their immune systems to recognize the bacteria, but without making them sick. They name it the Bacillus Calmette-Guérin vaccine, the BCG vaccine. The first patient is a baby in Paris in 1921, and from there the vaccine is rolled out wider. Not without some initial mishaps, but over time as manufacturing methods get better, it becomes safer and is used throughout the world. Today, 104 years later, the BCG vaccine is still being used. It's injected into the skin and results in a telltale scar on people's arms. In New Zealand, because TB faded out here, the BCG vaccine was removed from the national immunisation schedule in the 1990s. Today, it's only freely available for children under 5 years old at high risk of TB. But here's the thing. Despite it being the one vaccine against tuberculosis that we have that is used around the world, it turns out it's not super great at its one main job.
BCG has been introduced as a vaccine against tuberculosis, or TB. And while it has been in use for a very long period of time, unfortunately, the protection it offers is pretty low. So it can protect children, very young children, against tuberculosis that develop in other parts of the body except for the lung, what we call extrapulmonary forms of TB. But it doesn't protect adolescents or adults, unfortunately. But it is still the best thing we have in our fight against tuberculosis.
But because it's been used for such a long time, there are large populations of people that have been inoculated with this vaccine. And by studying them, some interesting things have become apparent.
So we have 100 years worth of data from the use of this vaccine in people. And what researchers noticed pretty early on, even back in the 1930s, was that individuals that received the BCG vaccine, they did have some protection against tuberculosis, but they were also protected against other things, notably being respiratory viral infection, but also sepsis, particularly in young children. So sepsis is basically an infection in the bloodstream. Fast forward a couple of decades, what people also found was that BCG was really good at stimulating anti-cancer responses. So it can provide this really broad level of protection, which goes against our thoughts when we're thinking about a vaccine providing specific memory-like responses in that adaptive arm of the immune system. We're getting this broad, non-specific response, which is more similar to what we would think of when we're thinking about the innate immune system.
Let's back up a bit for some Immune System 101, which we tend to think of in two different branches. First, the innate immune response,
which is a more sort of primitive, early, quick, non-specific response. And you see innate-like properties in lots of other animals, like in fish and birds, and even to a certain extent in plants and bacteria. So that's something that's been with us evolutionary for a long period of time. The adaptive side is something that's evolved much more recently, and you find this in mammals. And the adaptive side is much more specific, and so it's going to target protecting against very specific things like particular infections, but also really importantly is it has memory. So that's what we think about in our vaccines. We want to generate some sort of immune memory, and that's what the adaptive system does really well.
And that, of course, is how vaccines work. By introducing our adaptive immune system to a specific bug so that it can take a mugshot and work out a plan to defend against it if it ever sees it again.
We introduce something that mimics or looks very similar to a particular pathogen. And we can do this in a whole heap of different ways. So it can be something that is just a weakened form of the bug we want to protect against. It could just be a piece of the bug, a non-infectious piece of that bug, or we can even take the code for a piece of the bug, which is what we use in the mRNA vaccines, which we're all familiar with because of COVID-19. And so we can introduce this into the body. The body knows that this is something that's foreign. I need to make an immune response against that. And it will leave these long-term memory imprint and these memory cells that stay in your body for periods of years to decades. And so then when you see the infectious particle or the virus or bacteria again, the immune system goes, oh, hey, I know what this is. I can very quickly get that under control.
But as scientists learn more, the lines we've drawn between the innate and adaptive immune responses are blurring. Sometimes the innate side seems to show memory, and the adaptive side can protect in a general way. And of course, they interact with each other. In the case of the BCG vaccine, as well as triggering the adaptive side, it has an effect in boosting people's innate immune response, training it to be ready for other infections. And Kerry and her colleagues are keen to figure out how it does this. While they've not nailed it down exactly, they've got a few ideas as to what might be going on.
The first thing is that it is a live attenuated vaccine. So the immune system has more time really to interact with this vaccine because you're injecting a live bacterium. It has lots of different components. It's able to interact with the immune system in many different ways. The other thing that seems to be really important because it's live attenuated is it can travel to other parts of the body. So it doesn't just stay where you inject it. So people have found that you actually get changes within individuals' bone marrow, which is the factory where it produces all your immune cells. So you get injecting this bacterium into the skin, but you can get these changes happening in the bone marrow. And the really amazing thing about that is because it's this factory, all of the precursor cells that give rise to all your immune system, those can be altered. So you're altering something at a level that means you can have this long-term effect across your entire immune system.
And when they zoom in to try figure out exactly how it's doing this alteration, they think it's by epigenetic control. This regulates how tightly different parts of our DNA are folded. And tight folding means the machinery of the cell can't access a gene to read it.
What we think the BCG does is it goes and opens the DNA around areas that are really important for the immune response. So that means that cell can then go and use those genes, whereas previously it was not able to use them because it was all closed up.
The BCG vaccine is the only live attenuated bacterium vaccine currently used in humans. In animal models, other researchers investigated different live bacteria and saw signs of a general immune boost. So it's unlikely that this is specific to this tuberculosis bacterium. There are also live attenuated virus vaccines, for example, the polio and measles, mumps and rubella vaccines. And there's evidence that they induce a level of general immune protection in kids, but they've not been studied as closely as the BCG vaccine. It's different to actually catching the fully fledged virus or bacteria itself, says Kerry. They attack our cells and cause damage, and we don't see the same immune system training. Figuring out how it all works could help us make better vaccines.
When we think about a vaccine, it generally has a number of different components. One being what we call the antigen, so that is the main target that you want to focus your adaptive immune response against. The other part of a vaccine is something called an adjuvant, and this is something that provides a general immune boost. In a live attenuated vaccine, you basically get 2 for 1 because it all comes together. But for these more advanced vaccines, we have to separate out these components. And so in the ideal world, what we can do is we can take the lessons from BCG and feed that into a design of adjuvants or ways that we generally activate the immune system. And so that would provide that non-specific protection. So you end up with the best of both worlds. That's essentially what we are aiming towards.
Kerry studied the BCG vaccine during her time at the NIH, the National Institute of Health in the US, where she went on sabbatical, initially for 2 years, but this was extended when COVID-19 hit, and all of a sudden there was a bunch of research to be done. Back now at the Malaghan Institute of Medical Research in Wellington, Kerry and her team are further investigating some of these new findings. In the Tissue Immunity Lab, PhD researcher Bex Palmer explains what she's focused on.
I'm looking at understanding how our lung as a barrier tissue or organ responds to certain environmental insults. The main thing I'm looking at the moment is allergy and asthma and how that kind of immune response develops. It's kind of a defective immune response, so it's kind of coming up in response to something that's not harmful to us. So asthma is usually towards things that don't really cause danger, like pollen, house dust mite, peanuts even. And so understanding kind of the mechanisms which underpin the overall immune response, specifically in the lung.
Right, our immune systems are amazing and they're there to defend us, but sometimes they start doing things when they really shouldn't
Yes,
be, and that's the case
absolutely.
with asthma.
Yes. So I'm looking specifically at this fungal allergen, and it's known as Aspergillus fumigatus. So that's found kind of everywhere we breathe. It's quite present in the environment, but most of the time we don't experience this hypersensitivity to it. However, if you do have kind of predisposition to allergy and asthma, you can experience this hypersensitivity, and that can cause a lot of problems. And as we know, asthma is a high health burden in our public health system, and a lot of people suffer from it, and it's getting increasingly worse. So understanding kind of the mechanisms which drive this dysregulated response is really important for us to develop effective treatments.
When she says this fungus is everywhere, she means it. In the soil, in the air, and in our homes if we live in damp, mouldy houses. You can hear that loud hum in the background. That's because this lab is specifically set up for the team to work with infectious microbes or fungal spores to test how lung cells respond.
We've got a massive hood which is basically creates this air wall which allows us to work inside this space and not kind of let anything out. There's kind of this airflow that means that everything that's infectious is kept inside, and when we come out we're not bringing that infectious stuff with us. So there's a lot of different things that we work with in here that kind of helps us work safely. But in order to address these questions or understanding how asthma or allergy really affects our lung and our body, we use mouse models to kind of mirror what we would clinically see. And so we're using a low level of this fungus and we give them to the mice, and it's present in the airway. And then we can use these specialised methods like flow cytometry, which allows us to look at kind of the identity of each cell that we're interested in. And then we can kind of understand as a whole what kind of cells are present in the lung during different stages of this kind of sensitivity to asthma.
This will help Bex answer questions of which cells are involved in different stages of the immune response, and how this differs in these mice that show a hyperactive immune system asthma-like response. Bex will also use a method of single-cell sequencing to figure out what each cell's role is in the response. The big picture view, Kerry says, is that they're investigating how the immune system is being trained by every interaction it gets with a microbe, foreign particle, or vaccine.
So the BCG was a starting point for us to understand how this weakened vaccine interacts with the immune system. And so now we're taking that to the next step and say, okay, we know this weakened vaccine does this, but we're exposed to these kind of mild, non-dangerous microbes like all the time. We inhale them all the time. So our experience of that and our exposure to that really playing a role in the way our immune system develops in tissues. And so part of what Bex is looking at is particularly the role of the fungal spores, understanding how we interact with those on a day-to-day basis. Do we have sort of a mild response and everything's okay? Or what happens when you have an overreaction, an over-response, and the ultimate outcome of that is something like asthma? But one of the major reasons why we're paying close attention to this is because with climate change, the amount of aspergillus is increasing quite dramatically. So this is something when we're thinking about that balance between host and microbe and the levels of exposure going forward, we're going to be exposed to more and more.
And this team is interested in the lungs in particular, as the cells that line our lungs are often the first line of defence.
You're constantly being exposed, which is like, makes sense that there'd be kind of specific mechanisms within the lung that underpin kind of either rapid immunity or a rapid response, which is different to kind of what we would see elsewhere. But yeah, I just think that understanding more about how the immune landscape of our lung changes to to prior infections, and then how that determines downstream responses is so relevant.
Yeah, if you think of a human going through life, they're in a house that has fungal spores.
Exactly.
They've got the flu one year, they've got COVID-19 the next. Their lungs are consistently being challenged.
Yeah. And quite often we may have these mini responses in our lung that we don't even realize that it's happening. But, under the surface, there's this dynamic kind of interplay of responses that are kind of keeping everything at steady state or at homeostasis, which is just fascinating.
What it all comes back to is that balance between host and microbe, says Kerry.
Who's winning and who's losing at any point in time, and they change each other over the period of a human life. You see that the way in which your immune cells respond, the way in which they remember things they've seen before, really change the composition of the tissue as a whole. And that's why, particularly in this early life window, it's really important to understand what's going on, because when a baby is born, they're completely naive. They have some antibodies that they've received from their mother, which will protect them for the first few months of life. But then they start inhaling and being exposed to this big wide world. And the way in which those exposures happen and the way in which the immune system interacts with those different type of microbes could play a really important role in setting up what we call this tissue immune response going forward.
But back to tuberculosis. If the vaccine doesn't work all that well, where does that leave us? Well, it is caused by a bacterium, and we do have antibiotics.
But I think it's really important to note that this is not your standard antibiotic course that you take for a week and hope for the best. You have to take these antibiotics. It's a cocktail of 3, 4, 5 antibiotics depending on the strain of TB you have, but you have to take these for months.
Right.
Every single day for months, up to 6 to 9 months. And these antibiotics can make you feel pretty rubbish. So while we can cure TB, we're a long way from it being an easy task.
And now there are some strains that are becoming resistant to the antibiotics we have. So is there a way we can make a better vaccine? Well, there are different teams around the world working on that. But maybe there's also a way we can make the existing vaccine better.
What the field has found, including some of my colleagues at the National Institutes of Health when I worked there, they found that by giving the BCG intravenously, they could provide animals with sterilising protection against tuberculosis. And this is absolutely amazing. This is unheard of in TB research. And so what that tells us is BCG, it's able to do the job. We're just not giving it its best advantage. We're just not giving it the leg up it needs to provide the protection against TB. So the next step is how do we translate those findings that we see in animals to humans? And there's many different studies going on about how we could potentially do this by altering how the BCG interacts with the immune response. So this could be through delivering it directly into the lungs itself. So people would either inhale the vaccine or be squirted down their nose or something like that, or potentially looking at ways that we can provide a more sort of systemic protection through the bloodstream as well.
When it was first used, all the way back in 1921, the vaccine was given orally, and studies show this might be more effective. Even if it seems to work amazingly well in animals delivering the vaccine intravenously, that is, into the bloodstream, this is just not an option for the kind of worldwide vaccination program needed. Which is why researchers are investigating an inhaler or an intranasal method to get it to people's airways, the site of first interaction between cells and the bacterium. Interestingly, delivering the BCG vaccine directly to the site of action has been effective for something that Kerry mentioned right at the start, helping to fight cancer.
Early on, I think it was in about the 1940s, 1950s, they noticed that you could generate anti-cancer responses with BCG. And again, it came down to making sure the BCG can get into the right place to provide that interaction with the immune system. And so what was found was a really good model for this was actually bladder cancer. And how they would give the BCG to these patients is they instill it actually directly into their bladder. And the BCG is then able to stimulate an immune response that can go and kill off all of the cancerous cells. And it is still in use as the gold standard treatment for invasive bladder cancer.
I mean, do we know how that works?
We don't really understand why it works. It's one of those things people tried out because they'd seen an effect and we go, oh my gosh, this works. We're just going to keep doing this because we haven't found anything better. We think it's doing a couple of things. One, it's generally activating the immune system so that it makes it easier for the immune system to identify cancer as being bad and needing to be killed. But of course, the problem we have with cancer is that it's from our own cells. And so the BCG helps with that. But again, the other thing the BCG is doing is they'd found this in a mouse model of bladder cancer, is from the bladder, it can get into the bone marrow. And again, it reprograms those immune cells really early on in their development. So when they come out of the bone marrow and are circulating in the body, they're just much more efficient at doing their job.
For Kerry and her team, it all adds up to a complex and intriguing puzzle that they hope to pull apart. So we can learn how this immune system boosting works and use it to our advantage.
We can use BCG as a model to identify those factors that provide the non-specific protection, and we can then feed that into the design of future vaccines so that we can make the most of both the non-specific and specific protection.
Thanks to Dr. Kerry Hilligan and Bex Palmer from the Malaghan Institute of Medical Research. This episode was produced by me, Claire Kincannon, production help was from Ellen Rikers, and sound engineering was by Phil Bench. If you want to get in touch with us about the show, you can find us on Facebook at RNZ Science, or you can email us, ourchangingworld@rnz.co.nz. Tēnā koe I whakarongo mai. Thanks so much for listening. I'm Claire Kincannon. Kia pai, te wiki.